Hello everyone. In this video we will talk about a chapter, the fundamental unit of life. The fundamental unit of life. Now let us talk about some introduction. We know that every living organism starts its life as a single cell. Let me write this statement. Every organism starts its life as a single cell For example, if you talk about human beings, we know that humans are made up of several trillions of cells. 70 to 100 trillions of cells are present in human body, and here the human life actually starts with a single cell. Human life starts with a single cell, that is zygote, and we know that this zygote is formed by the fusion of male and female gametes in the fallopian tube or in the oviduct. Now here we know that this zygote is formed by the fusion of male and female gametes, and it is the single cell, and it is diploid in condition. And this zygote undergo several repeated mitotic divisions or cleavages, and it further developed into multicellular structure, and those multi-cells will be further developed into embryo, and that embryo further developed into fetus, and further development takes place in the mother's uterus. Now just let me write here, zygote which is formed by the fertilization process undergo several repeated mitotic divisions or cleavages, and it further developed into multicellular structure called an embryo, and this embryo further attaches to the mother's uterus wall, attaches to the mother's uterus wall, and we call this process as implantation, and there it further develops some extra embryonic layers, and it developed into a fetus, and complete growth and development take place inside the mother's womb. So a mother will give birth to a fully matured young one after completion of the pregnancy period. Now let us talk about organization of a living body. If you see the organization of a living body, so let us say human's life. Here the human life starts with a single cell, that is zygote, and that zygote undergo the cleavages. It is divided into multicellular structure, so that a group of cells are further divided into tissues, and group of tissues are further divided into organs, and group of organs are further divided into organ systems, and group of organ systems are further organized into an organism. So this is the organization of a living body. Now let us talk about the basic fundamental unit of life, that is cell. Here we can see that cell is the basic unit of all the living organisms. Here the cell is the basic unit of living organisms. Now let us talk about the definition of the cell. Here we can see that cell is the basic structural, functional and biological unit of life. Let me write here the definition of cell. Here cell is the basic structural, functional and biological unit of life. Here we must remember that cell is the basic structural, functional and biological unit of life. Now let us see here, there is a branch of biology that deals with the study of cell, and we call it as cytology. Here cytology is nothing but, we can say that it is a branch of biology that deals with the structure and study of the cell organelles. So actually the term cytology is derived from the two words. Let me write here the word cytos, which means cell, ology or logos, which means study. So simply we can say that study of cell structure and its organelles. So now let me write here the definition of cytology. It is the branch of biology. It is the branch of biology which deals with the structure and the study of cell organelles. Nowadays the term cytology is replaced with cell biology. Sir, what is cell biology? Here cell biology is also a branch of biology that deals with the study of structure, biochemistry, physiology, reproduction, evolution and genetics of the cell. First let me write the definition of the cell biology. It is the branch of biology that deals with the study of structure, biochemistry, physiology, reproduction, evolution, and genetics of the cell. Now let us talk about discovery of the cell. First, who discovered the cell, and discovery of different components of the cell and the different terms used by the different scientists. Now let me write here Discovery of cell. Here cell was first discovered by Robert Hooke. Robert Hooke was first discovered cell in the year of 1665. So here Robert Hooke, to observe the cell, he has taken thin slices of cork of oak tree, and he observed these slices of cork of oak tree under his self-designed microscope. Actually, Robert Hooke has invented his own microscope, and it was a double-lensed simple microscope. With the help of his double-lensed simple microscope, he discovered a cell. And when he observed that thin slices of cork of oak tree under his self-designed microscope, he noticed many honeycomb-like structures. Right? In that honeycomb-like structures, Robert Hooke noticed many empty boxes or many compartments. He called those empty boxes as compartments because all of those empty boxes are divided by the walls, and he named these empty boxes as cellulae. So later on the term cellulae is reduced as cell. Actually the term cell come from the Latin word cellulae, which means a little room or a small room. First look at here. In these two pictures we can see that this is the Robert Hooke's double-lensed simple microscope. With the help of this double-lensed simple microscope, he observed honeycomb-like structures, and in these honeycomb-like structures Robert Hooke noticed many empty boxes. Look at this. This is one box. Like this there are many empty boxes present, and he called them as compartments, and all of them are divided by the walls. Now let me write about discovery of Robert Hooke. Here in 1665, Robert Hooke observed thin slices of cork of oak tree. So now let me write here. Robert Hooke, to discover the cell, he has taken thin slices of cork of oak tree, and he observed these thin slices of cork of oak tree under his self-designed microscope, and observed under his self-designed microscope. Under his microscope, he noticed honeycomb-like structures, and he called them as empty boxes or compartments, and all these compartments are divided by walls. So here Robert Hooke named these empty boxes as cellulae. So later on the term cellulae reduced as cell. The term cell came from the Latin word cellulae, which means a little room or a small room. So this is, we need to remember, the term cell came from the Latin word cellulae, which means a little room or a small room. Robert Hooke has written a book called Micrographia. He wrote a book called Micrographia. So about his discovery, everything was mentioned in this book, Micrographia. So here actually Robert Hooke discovered a plant cell, and is it a living cell or dead cell? It is a dead cell, a dead plant cell. So now just let me write over here. Here in the year of 1665, Robert Hooke observed dead cell which is resembled honeycomb like structure. Honeycomb like structure. So here we can see that actually the cell discovered by Robert Hooke was a dead plant cell. So just let me conclude here. Robert Hooke actually discovered dead plant cells. Then first, who discovered living cells, sir? Now let us discuss about a scientist first who discovered living cell. His name is Anton van Leeuwenhoek. Anton van Leeuwenhoek. In the year of 1674, Anton van Leeuwenhoek discovered living cells in pond water. That means he has collected a sample from the pond water, means little water was collected, and when he observed it under his self-designed microscope, he noticed some unicellular organisms like amoeba and paramecium, and we know that they belong to a group protozoa. So here Anton van Leeuwenhoek discovered protozoans. The name of these protozoans: amoeba and paramecium. So actually Anton van Leeuwenhoek called these organisms as animalcules. He called these organisms as animalcules. Later on the term protozoans was given by some other scientists. And he has also observed bacteria in tartar of teeth. So look at this. He is the Anton van Leeuwenhoek, first who invented single lensed simple microscope. See, this is the single lensed simple microscope. With the help of this single lensed simple microscope, he has observed protozoan organisms like amoeba and paramecium, and he has also observed bacteria by collecting the yellowish colored part which is present on the teeth. And when he observed it under his self-designed microscope, he noticed certain bacteria. And he has also observed sperm cells in human sperm cells. He has also observed sperm cell in humans and red blood corpuscles. He observed red blood corpuscles in fish. So here our doubt is clarified, that is the first cell was discovered by Robert Hooke and the first living cell was discovered by Anton van Leeuwenhoek. So the cell discovered by Robert Hooke was a dead plant cell, whereas living cells was discovered by Anton van Leeuwenhoek. And there are some other scientists who gave some specific terms to the living fluid present in the cell. Here there was a scientist, name of the scientist is J.E. Purkinje. J.E. Purkinje called the fluid part inside the cell as protoplasm. So he has coined the term protoplasm. Coined means he has given the term protoplasm to the living fluid inside the cell. So we generally call the living fluid inside the cell as the cytoplasm, and cytoplasm and nucleus, these two together called protoplasm. Actually the term protoplasm was given by this scientist J.E. Purkinje. He has coined the term protoplasm. Now, so let us understand what is protoplasm. Here protoplasm is nothing but cytoplasm and nucleus. These two cellular components together called as protoplasm. So what is protoplasm? It is a living fluid present inside the cell. And later on there is another scientist, Huxley, called this protoplasm as a physical basis of life. Physical basis of life. Because the genetic material present in this protoplasm, which is responsible for the formation of life, hence he has called it as a physical basis of life. And the important component of the cell, nucleus, it was first discovered by Robert Brown. Here we need to remember the two terms. One is Robert Hooke, name of the scientist. Robert Hooke discovered a dead plant cell, whereas Robert Brown in the year of 1831 discovered a nucleus in orchid leaves, and inside the nucleus there is an important component which is responsible for the RNA synthesis. So that is nucleolus. Nucleolus was discovered by Felix Fontana. It is easy to remember, name of the scientist Felix Fontana. He has discovered nucleolus, right sir? And to observe the cell and different components of the cell, which device is required? Yes, we need a device, that is a microscope. Now first let us discuss about microscope and its types. So first, what is microscope? Microscope is a device which is used to observe microscopic structures, which is used to observe microscopic structures which we cannot see with our naked eye, which we cannot see with our naked eye. Naked eye means unaided. Now first let us talk about the types of microscope. Types of microscope. Here microscope is of majorly two different types. This is on the basis of presence of number of lenses. Simple microscope and compound microscope. So just now we discussed different scientists discovered different components of the cell with the help of their own microscopes or self-designed microscopes. Now let us talk about the first microscope which was used by Robert Hooke was a double-lensed simple microscope. Double-lensed simple microscope. It was invented by Robert Hooke. And the other one, single lensed simple microscope. It was invented by Anton van Leeuwenhoek. Now see here, these are two, double lensed simple microscope and single lensed simple microscope. Both are type of simple microscopes. So how can we say these two are simple? Because here the number of lenses are 1 or 2. If any microscope, if it possesses 1 or 2 objective lenses, then it is called as a simple microscope. Whereas in compound microscope, where more than 2 objective lenses are present, and here the compound microscope was invented by Janssen. Zacharias Janssen. Here the simple microscopes are of two types, double lensed simple microscope and single lensed simple microscope. In double lensed simple microscope, we have seen that 2 objective lenses are present. So over here, so this is one lens, this is one lens, there is another lens present in this. So that is here glass lens, and this is the another lens. So here 2 lenses are present. Whereas in single lensed simple microscope, here there is only a single objective lens is present. But in case of compound microscope, more than 2 objective lenses are present, and it was invented by Janssen or Zacharias Janssen. Now let us talk about another important type of microscope, that is electron microscope. It was first invented by the 2 scientists Knoll and Ruska. in the year of 1932 they first invented this electron microscope. It is used to study the ultra microscopic structures. See, actually it was first invented in the year of 1932, but it was first used in 1940. And there is another important microscope, that is phase contrast microscope. Phase contrast microscope. It was invented by Jernaik, and it is used to observe moving cells. So that is, we can observe cell division, and it is also helps in to see the unstained cells. And with the help of this electron microscope we can observe the ultra microscopic structure of a cell. That means inside the cell organelles, internal structures can also be seen with the help of electron microscope, whereas under phase contrast microscope we can see moving cells and also we can see unstained cells. Now first let us talk about structure of compound microscope. Here under this we can see the microscope structure. Look at this picture. Here we can see this is the structure of compound microscope, which is first invented by Z. Janssen or Zacharias Janssen. Now first let us talk about the different parts present in this compound microscope. The first, let us talk about this base. Weight of the microscope is held by the base. Okay. The next, the important part, this is called arm. Arm. Arm is used to hold the microscope. Just let me note down. Here base is the important part, and the next one is arm. With the help of this arm we can hold the microscope. And another important part of the microscope, see, this is body tube. Body tube. In this body tube, a draw tube. See, this is the draw tube. This is body tube, and this one is draw tube. And another important point of this compound microscope is lenses. Here in this compound microscope, two type of lenses are present, and these two type of lenses are, one is present at top of this draw tube. So this one, this is eyepiece lens, which is also called as ocular lens. Eyepiece lens, also known as ocular lens, through which we'll see the object which is placed on the stage with the where we already prepared a temporary slide. And the another type of lens is present at lower part of this body tube. In the lower part or posterior part of this body tube, we can see that there is a nose piece, this round one, this is nose piece, and to this nose piece 3 objective lenses are present. So in compound microscope we can see the 2 type of lenses. One type of lenses or eyepiece lenses are also known as ocular lenses, and the another one is objective lenses. Here more than 2 objective lenses are present with different magnification powers. Now let us see here the magnification power of eyepiece lenses ranges from 5x to 10x and 45x, whereas the magnification power of objective lenses would be also 5x. Its range is from 5x to 10x. It will also be 45x and 100x. Not all the objective lenses with the same magnification power, they are with different magnification powers, right? And another important part of this compound microscope is the stage. Just below these objective lenses you can see that there is a square shaped structure, stage. Here on the stage clips are present, so we can call them as a stage clips. Stage clips. These 2 stage clips are present. See, this is one stage clip and the another side there is one more is present. These 2 stage clips are used to hold the glass slide. Hold the stage clips can hold the glass slide. Next, just below the stage there is another important point, so we call it as condenser. In this condenser we can see there is a presence of diaphragm lever and there is a slit present which is passes through this stage. Below this condenser a mirror is present. So this mirror acts as a reflector. It acts as the reflector. So whatever the light fall on this mirror, that will be reflected and that passes through the slit present in the stage. And after preparation of the temporary slide or a permanent slide, if we wanted to observe any plant cell or animal cell or any other objectives that we need to prepare a slide, or permanent slides are also available. So when you place it on the stage, see with the light which is reflected by the mirror will passes through that material which is placed on this glass slide. So that image can be seen with the help of these objective lenses and eyepiece lenses. That image will be formed in this body tube. Now let us talk about the important parts of this compound microscope. The coarse adjustment, this is rotated in both the directions. So when we are rotating it, the entire body tube along with the draw tube and objective lenses can move up and down. So here this can be moved to get the desired focus. To get the desirable focus we will rotate this, whereas the fine adjustment can also be rotated in both the directions. This can be rotated in both the directions to get the sharp image of the material whatever we have taken on the glass slide. Just let me note it down. The coarse adjustment is used to get the desired focus get the desired focus, whereas fine adjustment is used to get the sharp image, to get the sharp image. Now let us talk about working mechanism of compound microscope. The working mechanism. Whenever we wanted to observe any plant or animal cells, so we have to prepare a temporary slide, and when you place it under compound microscope, we have to fix this on the stage like this with the help of stage clips, so that the glass slide cannot move here and there. So here when the light is fall on this mirror, the light will be reflected and it passes through the material which is kept on the stage, and here with the help of objective lenses and ocular lenses we can see the image. So here actually the real and inverted image of this material which we have taken on the glass slide is formed by the objective lenses. So here objective lenses forms the real and inverted image, and it is further converted into virtual image by the help of these eyepiece lenses. So this is the working mechanism of the compound microscope. Now just let me note it down. The real and inverted, the real and inverted image, the real and inverted image of the object formed by the objective lens is changed into an erect and virtual image, an erect and virtual image by the eyepiece lens. So this is about structure of compound microscope and its working mechanism.